Hi everyone, welcome back. So, there are very few places on Earth that are actually devoid of bacteria. There's a lot of different species out there, and unique species of bacteria can be found in incredible places, like in the deepest reaches of the ocean, circulating in the atmosphere, in every desert and frigid pole of the planet, on the surface and on the inside of every animal and plant and fungus. Even buried deep in rock and ice and in the soil and everywhere. There are very few places that are actually devoid of that life. And so we think of bacteria as ubiquitous in our daily life, as in they're found everywhere all the time. In fact, bacteria are incredibly challenging to keep out of places. There are lots of places that we want to maintain sterile conditions, but we actually find that we can't. We've realized that we've actually accidentally sent bacteria to the moon at this point, and that's despite a lot of effort that's gone into keeping those environments incredibly sterile, specifically out of concern for possibly, taking bacterial life to a place where it was not originally there, and possibly confusing people in the future if they ever go to the moon looking for life, for example. So it can be very difficult to keep them out of places. And while bacteria generally are everywhere, despite their ubiquity, the nature of existing in all these different places, species are often actually limited in where they can grow. So bacteria are everywhere, but specific species are not. The bacteria that grow on hydrothermal vents in the ocean are not found anywhere else naturally. They live off of the rare resources like hydrogen cyanide that are found in those vents. That's a resource that we can be thankful is not, anywhere else, like in our normal environment, because that would be toxic for us. Those bacteria require those resources, and so they live there so they can utilize them. They evolved to live in these extreme conditions, in these intense, fluctuating temperatures that, in one area it'll be near freezing, in the other it'll be over boiling, it'll be extremely hot, these volcanic gases are being emitted. And so, these are conditions that are not found everywhere. They live there because that's where the conditions are. If we took these species and we brought them to, say, our backyard, they most likely wouldn't be able to survive because they wouldn't have those important conditions that they need to survive. And they would also, if they could survive those conditions, they would probably be outcompeted and eliminated by our, competitors, species that do live in these, our backyard, and that's their native environment. And so they're better at surviving under those conditions. So they would outgrow and eliminate these introduced competing species. In many cases, this is because of those specific habitat demands for those bacteria. They're dependent on those specific conditions. And if they aren't getting those conditions, they're not going to be able to grow, to reproduce, and might actually be killed off because of the lack of those conditions. Other times, competition with other microbes is really important because these species, they're not all equal in their ability to survive under certain conditions. Some species are going to be much more efficient at surviving under those conditions. And a history of evolution, growing and living and reproducing in that environment can select for features and characteristics of that bacteria that make them, all the better at surviving under those conditions. Species that are not from that environment might find it more challenging, and so they can be outcompeted by these native competitors. So these species can vary a lot in the conditions that they require. There are some species that are very needy. We call them fastidious. Fastidious. It means that they have they're very picky. They require rare resources or specific conditions that are not present everywhere. And then the not-so-needy species, sometimes we call them generalists, but the proper term is non-fastidious. Non-fastidious. So leaving their natural habitat sometimes means potentially being less able to survive in those new conditions wherever they end up, either because those conditions they go to are less ideal or because there are competitors in that new environment that are just better than them at surviving. And remember, few places in this world are truly devoid of bacteria. A moving microbe, when it goes to a new place, it's rarely ever going to find a place that is sterile, that is without other life. They're going to be moving into a place where there are already residents. And if those residents are better competitors than the new bacterium, as in they're a better fit for that environment, the new bacterium is going to struggle. It's going to be less effective at finding resources, at avoiding threats, at growing, at reproducing, and so on. And so the better competitors are going to be the ones that stick it out. When we discuss microbial growth, we're not really talking about an increase in the cell's size. Although that can happen, that's not what we're talking about. Really, we're talking about an increase to the colony's size, the growth of the population, okay? Growth that specifically occurs through reproduction. So the result of this, the result of bacterial growth is a discrete colony, like what we talked about previously, a single cell. When it divides and divides and divides and divides, it will form a distinct colony. And a colony is just the aggregation of cells that arise from a specific parent cell, like an original ancestral cell. And so when we grow bacteria on plates like this, when we have distinct dots where bacteria are highly concentrated right there, that single dot, that arose from a single cell that was placed at that location, thanks to this person's excellent streak plating method. And so as a result, you had a single cell that landed here, and it was able to reproduce unabated, and eventually became a whole colony of millions and millions and millions of cells, all packed together in this same little area. Now, bacteria reproduce asexually, if you remember. They don't need a partner to do any of this. So they're not limited by like a difficulty of finding a mate or anything like that, like animals would be, for example, because animals are mainly sexual reproducing organisms. We need another partner in order to make another human, for example. So if you don't find another partner, you're not having another human. Not without science getting involved. But bacteria are not bothered by that. They can just divide their cells. So most bacteria reproduce by something we call binary fission. It just means that the cell, it simply divides in two. One cell splitting into two. It's a very simple form of cell division. Other species can engage in more complex forms of fission, like non-binary fission. So this is when one cell divides into more than two cells, like 4, usually, things like that. In all cases, the parents and the descendants are genetically identical. There's no new DNA that's come into the picture unless there's been a mutation. Not like the way that we reproduce. We have a partner, and we get DNA, half of the DNA that becomes part of the new person that is created. Comes from one parent, and half of it comes from the other, and so that person, the offspring that forms, it's a mix of DNA that is yours and DNA that is not yours. It's 50% new DNA, quote unquote new DNA. Now, we're going to talk about doubling time as well, an important term. Okay, doubling time. Another way to say this is generation time. This is another way of talking about the rate of microbial growth. the average time that it takes a bacterial cell to divide, okay, to essentially create a new generation of cells. Now, all cells in the colony are similarly dividing. They're all going to be reproducing like this, okay? They won't all be on an identical timeline. They'll be staggered, right? But they will all be, working to the same goal, essentially, to reproduce and reproduce and reproduce. Now, because these bacteria can divide into two, for the species that do binary fission, the population can grow really rapidly. We can see what we call exponential growth, at least for a time. So in the case of binary fission, this is pretty straightforward. One cell divides into two, and in the next generation, those two cells divide to make 4 cells. The next generation, those 4 cells divide to make 8. Then those 8 cells divide to make 16, and 16 to 32, 32 to 64, 64 to 128, and within a really short amount of time, you're looking at hundreds, thousands, millions, billions of cells, crazy amounts. This exponential growth can be even greater when we're talking about non-binary division. So like when one cell divides into 4, and then the next generation, those 4 cells divide into 16 cells, and so on. But in any case, the colony is going to be swelling as the population grows. It's going to grow and grow and grow. Now, this growth, it's not actually exponential forever. And in fact, there's a specific pattern that emerges as bacterial colonies grow. At the very start, there isn't even really significant growth. We start when a new, imagine that you've inoculated a plate. Or, in another setting, you could have just a single cell. A single cell makes its way to a new habitat. Okay, it doesn't have to be in a laboratory setting. But the bacteria arrives in a new habitat and begins to look to grow and reproduce there. Initially, there's something we call a lag phase. Okay, a lag phase. So in this lag phase, bacteria it's not focusing on reproduction. Instead, it's focusing on acclimation to these new conditions that it's arrived at, and resource gathering to make sure that it has what it needs to survive. After we get to that point, and we cross that threshold where the bacteria feels acclimated to this new environment, you start getting growth, you start getting reproduction, you start getting the exponential growth that we talked about. That is what we call the log phase. In this phase, the birth rate is much greater than the death rate. As in, there are more cells being born than there are cells dying. And now, this graph, the way that I've displayed this, it looks very linear. It's like straight line here, but this is kind of misleading. This is a logarithmic adjusted graph. So, what we're really looking at is a graph that looks like, we have growth, exponential, 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 and it shoots straight up like that. But this graph has been adjusted to essentially simplify that. Okay. During this time, you've got doubling and then doubling those doubles and then doubling the doubles of those doubles and so on, right? So you've got rapid growth. The population, the colony swells massively during this time. Now, why do these bacteria lag before they log? Well, the reason why is it really comes down to adaptation, okay? These bacteria need to acclimate to those new settings, because they have to adjust their behavior. Bacteria have all these different proteins they can make. And they can make these proteins and in various amounts. They can make some, they can make none, they can make a few, they can make some, they can make a lot of all of the different proteins that they have. But those proteins aren't all useful to them all the time. These bacteria have to adjust their behavior, as in which proteins they make and when and how much. They have to adjust that to the specific conditions of their environment. This quality, this flexible quality of bacteria, we call this plasticity. We can say that bacteria are plastic in that their characteristics and their behavior, as in, which genes they express, which proteins they make, is somewhat flexible. You can adjust those things. You can alter them as you need them. These environments that they're going to be living in, the conditions they live in, they're going to change and differ, by geographic space, like where they are, and also by time. They can just change over time, and so bacteria can't behave identically under all conditions. They have to adjust and change their behavior as the situation demands. Okay. Now, from our perspective, we could talk about it like this. Imagine that you got put on a plane and had to suddenly move to some place that was much hotter than Michigan, like Rio de Janeiro. Okay, you go to Brazil. All right. Now, I'm confident that we could all survive in Brazil. Brazil is a wonderful place. I think it's a gorgeous place, but it's not the kind of thing that we could just like pick you up and drop you there, and you would necessarily be well equipped to survive right away. We have to adjust our behavior in order to live in that new environment. If we move to a place that's a lot hotter than Michigan, for example, now you have to adjust your behavior. You have to drink more water. You have to wear fewer layers. Maybe you spend more time in front of a fan or with the AC on and things like that. This is what bacteria are doing too. They move to a new place and they have to evaluate their conditions and their environment, and they have to change their behavior, their gene expression accordingly. Now, this doubling time, during the log time, during this log phase of microbial growth, doubling time is really going. You've got bacteria doubling and doubling and doubling and doubling and doubling. Well, the time that it takes for these bacteria to double can vary a lot by species. Okay, some species are going to double a lot faster than others. And this is something that can be as diverse as bacteria themselves. Okay, so species like Vibrio natrigens. This is a species that is, I believe this is a species that's like an ocean, a free living ocean species, free living halophile. And so this is a species that has a really fast doubling time. It's the fastest known to our knowledge. It can double in less than 10 minutes, right? And then you have, on the other end of the spectrum, you have species like Mycobacterium leprae. So this is the species that's responsible for leprosy. It's also pretty closely related to the species that causes tuberculosis, Mycobacterium tuberculosis. This is a very slow-growing species, so its doubling time is quite slow. It ranges between 12 to 14 days. Both of these numbers, all these numbers are assuming ideal conditions for growth. So under the best possible conditions, this is what we see. Now, resources, though, are finite, okay? So exponential growth can't continue forever because eventually there will not be enough resources that are available, or space, which is itself a resource, for these species to survive all in the same place, okay? So eventually growth is going to slow. Growth slows as bacteria run low on these unexploited conditions and low on space, room, like physical room. And so eventually the population is going to taper off. It's going to slow its growth and become stable, reaching something that we call the carrying capacity. That is the maximum number of organisms that a habitat, that particular space, can support on its own. Okay. Now we call this the stationary phase. Okay, that tapering off and eventual rough stability. It isn't exactly truly stable. We see here that birth rate is equal to death rate, but it's not really always exactly equal. Sometimes you get these oscillations that happen because the population grows and then some individuals begin to starve because they don't have access to all the resources. So then death rate picks up and it drops. And then some species, the effects of that starvation still linger, and so they can still be dying. but then you're under the carrying capacity, and so growth rate picks up again. And so you can have this oscillation like this as the population kind of circles around that carrying capacity. Okay? So this is a very important phase, and this can go on for quite some time, the stationary phase. Over time, inevitably, this is going to change, though. Resources are going to decline because bacteria are taking up a lot of those resources, and they're putting them into their body. In the process, they're using them and turning them into resources that, they're not gone, they're just changed, put into a different form inside of the body of these bacteria that is not necessarily accessible to other bacteria, not at that time. And at the same time, waste products are also building. That by the nature of bacteria living, by the nature of any organism living, they're going to be creating waste products. Lots of things that we make, but as a byproduct, as a waste product of some other process. And a lot of these waste products can be toxic to the bacteria or other organisms. And eventually, these conditions can cause the population to decline. It can shrink by a lot as the birth rate becomes less than the death rate. More organisms are dying than are born. And that population can decline, but rarely ever really going to zero. It's likely that the bacteria will still keep on circulating and surviving in these areas, even though the resources become less available. They don't really disappear. Again, they just become less available. And so the carrying capacity can also shrink like this. For less free resources ultimately means that the carrying capacity becomes less. The environment can support fewer organisms. We actually see this pattern, this lag, log, stationary death phase in a variety of organisms, a variety of microbes. We see this a lot with yeasts, and we see this pretty significantly in when we brew alcohol. And we can really see this. So yeasts are responsible for this, for our brewing of these of alcohol. So this is, of course, how we make beer and other kinds of alcoholic drinks. The yeasts are actively fermenting sugars that are in these big vats. They use that for energy. And in the process, they make these toxic byproducts, mainly alcohol, ethanol, okay? That this is the byproduct that is made. It's not the intended thing. The yeasts are not benefiting from making alcohol. It's a consequence of them using sugars for energy. And so it's toxic. It's really bad for these organisms to be surrounded by alcohol, right? It's toxic for them. It's toxic for us. It's toxic for most life. And so as these yeasts grow and they become more abundant, and then they hit the stationary phase and they survive in this environment for some time, they're making more and more and more of that alcohol. This is great for us who want to brew beer, but it's really bad for the yeasts because they're not able to get rid of that alcohol. It's being concentrated in that environment, and so that can also lead to the population's decline. In fact, it is possible in these situations for alcohol to be so abundant, for the yeasts to be so productive for alcohol that they can actually poison themselves and destroy their own population. This doesn't happen all that often in nature. It's something that we make happen in essentially laboratory conditions when we brew beer. But this is a pretty interesting example of that death phase in action.